Hello everyone. Today I will be discussing about the application of the cloning. So, let us begin with the topic. Genetic cloning, a key technique in biotechnology, involves the creation of genetically identical copies of an organism, gene or cell. This process has significant application in various fields and offers numerous advantages that have revolutionized scientific research and practical applications. Here we will explore application of genetic cloning and its advantages. It is a technique used to create exact genetic replicas of genes, cells or animals, or a method of producing identical genes through different procedures is known as cloning. Some clones already exist in the nature. For example, bacteria make exact copies of themselves each time they reproduce. In humans, identical twins are similar to clones. To clone a gene, DNA from a living creature is taken and inserted into a carrier like bacteria or yeast. Every time that carrier reproduces, a new copy of gene is made. There are three different types of artificial cloning: gene cloning, reproductive cloning and therapeutic cloning. Gene cloning produces copies of genes or segments of DNA, whereas reproductive cloning produces copies of whole animals and therapeutic cloning produces embryonic stem cells. Genetic cloning allows scientists to isolate and clone specific genes, enabling them to study genes expression and regulation. By introducing cloned genes into different host organisms, researcher can understand how genes function and interact in different genetic backgrounds, providing valuable insight into various biological processes. Cloning has been applied in animal biotechnology and plant biotechnology to produce genetically identical animals and plants respectively with specific traits. This technique has been used to clone livestock animals for improved breeding, conservation of endangered species and even the creation of transgenic animals that produce valuable pharmaceutical in their milk. Cloning help in the insertion of desired genes into host organisms, leading to the production of important compounds on a large scale that we can see here in case of crop breeding, in environment, in biopharmaceutical and in growth hormones. Some of the significant applications of cloning include: first, reproductive cloning. This involves creation an identical copy of an existing organism. Reproductive cloning can be used to preserve endangered species, produce genetically superior livestock and replicate pets. Second is therapeutic cloning, also known as somatic cell nuclear transfer SCNT. Therapeutic cloning aims to create embryonic stem cell that genetically matches with a patient. These cells can be used for potential regenerative medicines to treat various diseases and injuries such as diabetes, Parkinson's and spinal cord injuries. Third is research and medicine. Cloning plays a crucial role in scientific research. It enables the production of identical organism for studying genetic factors and disease mechanism, as well as testing potential treatment and drugs. It also helps in understanding embryonic development and gene function. Fourth application is agriculture advancement. Cloning can be applied to produce high quality plants and animals with desirable traits such as increased resistance to disease, improved yield and better nutritional content. This can significantly impact agriculture and food production as well as address food security challenges. Fifth one is organ transplant. Organ transplantation is a challenging medical field due to organ scarcity and the risk of rejection. Cloning offers the potential to create organs that are genetically identical to the patient, minimizing the risk of rejection and the need for immunosuppressive drugs. Sixth one is conservation efforts. Cloning can aid the conserving endangered species by creating genetically identical copies from preserved cells or tissues. This approach can help restore populations that are risk of extinction. Its application in drug production also. Cloning can be used to produce large quantities of specific proteins and antibodies for medical and research purpose. It offers a cost effective and efficient means of manufacturing these substances. Another application of cloning is gene banking. Cloning can be used to preserve genetic material from endangered species or individual for future use or research. In biopharmaceuticals, DNA cloning is used to produce human proteins with biomedical applications. One example is the production of insulin using E. coli. Other examples includes human growth hormones. Next application is gene therapy. Gene therapy is used to provide a specific gene that is not available in a patient's body due to a genetic disorder. DNA cloning has been used to build plasmids containing a normal version of gene that is non-functional. Next application is gene analysis. DNA cloning has been used to create artificial recombinant versions of genes that help them understand the function of that specific gene and how and it functions in an organism. Then genomic library. Cloning is also useful for identification specific gene which can be multiplied by growing relevant clone in culture and lead to formation of genomic library. Next application is stem cell. Stem cell can be produced through a technique called somatic cell nuclear transfer SCNT, which is a type of cloning. SCNT involves transferring the nucleus of a somatic cell, any cell other than a sperm or egg cell, into an egg cell that has had its nucleus removed. This process results in the creation of an early stage embryo which contains embryonic stem cell that are genetically identical to the donor of the somatic cell. Next one is less chances of immune rejection. Stem cell which are generated via SC and T have the identical genetic makeup for the patient to be treated, thereby reducing the chances of immune rejection by the recipient's immune system. Next application is increased knowledge of development and differentiation. Researchers are gradually gaining insight as to how to development of organism occur from single cells and how the birth defect arise. Consequently, they might learn how to control differentiation of stem cells, thereby producing a specific tissue. It might help in understanding the abnormalities in differentiation, wherein such abnormality might lead to diseases such as cancer. Next application is DNA fingerprinting. It is a laboratory technique used to determine the probable identity of a person based on the nucleotide sequences of certain regions of human DNA that are unique to individuals, example in forensic science. Next is mutational studies and identification of mutation. Helps to determine the presence of mutations as well as other studies on mutagenesis. However, it has been substituted with the polymerase chain reactions technique. Next one is gene function and structure. The structure or sequence of a particular gene can be identified by the process of cloning. An important gene can be identified and sequenced to determine the variations in sequence within. Next application is it prevent extinction of a certain species. Cloning appears to be a promising approach to restoring populations, as many organisms are near endangerment and extinction. Cloning can even help increase the diversity of genetic traits. Next application, it enables same gender couples to have offspring. Cloning would enable same gender couples to conceive a baby, except for the first fertilization procedure to make the embryo. Direct insertion of a adult somatic cells in a woman would not need a male. Next application is in a disease modeling. Genetic cloning enables the creation of animal models that mimic human diseases, aiding in the study of disease progression and testing potential treatments. These models provide invaluable insights into diseases like cancer, neurodegenerative disorders and cardiovascular conditions, leading to better drug development and treatment strategies. Next application of cloning is reproducibility. Genetic cloning ensure the production of genetically identical copies, allowing researcher to perform consistent and repeatable experiments. This reliability of essential in obtaining accurate and meaningful result in scientific studies. Next is targeted genetic modifications. Cloning facilitates the precise transfer of specific genes or DNA sequences into host organism. This level of targeted genetic modifications enable the creation of organism with desired traits and characteristics benefiting agriculture, medicine and other field. Next application in the preservation of genetic traits. Cloning allows the preservation of valuable genetic traits in plants and animal both. It is particularly crucial for conserving endangered species and maintaining genetic diversity. Next is personalized medicine. Therapeutic cloning holds promise for personalized medicine by providing patient specific stem cells for regenerative therapies, reducing the risk of rejection in transplant procedures. Last application is understanding genetic basis of diseases. Cloning animal models that carry specific genetic mutations helps researchers understand the underlying mechanism of various diseases and aids in developing targeted treatments. It is essential to note that cloning technology raises ethical and moral concerns, particularly when applied to human cloning. Many countries have laws and regulations governing the use of cloning technologies to address these concerns and ensure responsible application. In conclusion, genetic cloning plays a pivotal role in biotechnology, offering a wide range of application. Its ability to create genetically identical copies, modified genes and produce valuable compounds has significantly advanced scientific research and opened doors to various practical application in medicine, agriculture and environmental conservation. As technology continues to progress, the potential of genetic cloning in biotechnology is likely to expand further, leading to even more groundbreaking discoveries and innovation. Thank you.